In Chapter 10, we're going to focus on cellular reproduction. So you think reproduction and you might think sexual reproduction, which will be part of this, but we're also just talking about cell divisions. So you have a skin cell and it divides into two skin cells. That's a form of cell reproduction. There are different types of cell divisions in eukaryotes that we will talk about. We'll also talk a little bit about prokaryotes. We're going to talk a lot about the cell cycle and control of the cell cycle, because control of the cell cycle is very important when we talk about the concept of cancer. Cancer is unrestricted cell growth, basically. So when the cell cycle breaks down, we often see cancer. We will need to talk a little bit about the genetic material in this section, because when a cell divides, the genetic material needs to be duplicated faithfully. So we'll talk a little bit about chromosomes as well. So one of my favorite questions is, how do multicellular organisms go from the single-celled stage, which is called the zygote, to their complicated life form? And different organisms have different mature life forms. We have something here like a sea urchin. Here we can see the very first cell division from the zygote becoming two cells to maybe this, I don't know, it's probably a blastocyst of some sort. I don't know my sea urchin development very well, but then they go into this large structure that's many, many cells with their shell and their spines, and inside of it they have their soft, fleshy tissue. Sea urchin development actually will come up in this series of chapters, not because sea urchins are cool. They are cool, but because sea urchin cells were very easy to observe. And particularly when we talk about chromosomes, they had very clear cells where we could actually see the chromosomes inside of them. So they're very important, actually, in these coming chapters. But think about yourself, right? You start as a single cell and you become you. And we see development occurring there, right? You have lungs and you have skin and you have hair and all these things. How does that happen? It's coming from cell division. So in chapter 10.1, we're going to talk about the basics of cell division. We need to talk about some of the structures that are in here, particularly the genome. So we'll talk about prokaryotic genomes and eukaryotic genomes. We'll talk about chromosomes, the portions of the chromosome that we call genes, and we'll talk a little bit about this term trait, which will be important in the genetics chapter. Then we're going to talk about how chromosomes basically fit inside the nucleus and compact themselves during division. So we will start with prokaryotes, but then we will talk a lot about eukaryotes, where of course we have the nucleus there that is also involved in this process. So let's get into it. All right, genomic DNA in prokaryotes. So let's define the genome first. The genome is all of the DNA found within a cell, okay? In a prokaryote, that's made up of a single circular chromosome, and it might have some extra little bits called plasmids. Those extra circles of DNA called plasmids, kind of cool because they can be shared from one cell to another. Now, that's really of interest when we talk about things like antibiotic resistance, because one bacteria could have antibiotic resistance on its plasmid, and it could share that with another bacteria. It could even share it to another species of bacteria. So think about that for a moment. That's called horizontal gene transfer. That's like you giving some of your genes to your cat, or your dog giving you some of its genes. That's weird, right? We don't think about that in our world, but bacteria are doing that all the time. So these plasmids are really cool. And if you're interested in molecular biology, biotechnology, plasmids are very, very useful. We'll talk about some of their uses later in this course. Okay, so in bacteria, we have the circular genome. I swear this is a circle if you stretch it out. And then these small plasmids that can be in there. That could be the genome of the cell. In eukaryotes, things are a little more complicated. In eukaryotes, we have multiple linear chromosomes. They're double-stranded DNA, just like bacteria, but instead of being circles, they are linear. And you might have one or more different versions of this genome in there. Now that's really important, and we have to talk about this topic called ploidy. So the ploidy is the number of copies of the genome in the cell. What do I mean by that? Well, In your cells, you have 23 different types of chromosomes. Okay, chromosome 1, 2, 3, 4, 5, 6, 7, all the way down to the sex chromosomes. You are a diploid, which means that each of your cells has 2 copies of the genome in the cell. So you have 2 copies of chromosome 1, 2 copies of chromosome 2, so on and so forth. Diploid, 2N, 2 copies of each chromosome. One of those is coming from the maternal lineage, and one of them is coming from the paternal lineage, okay? I'm going to use those terms here instead of mother and father, because mother and father has social connotations to it, right? The maternal lineage is the one that donated the egg, and the paternal is the one that donated the sperm, in the case of human reproductive biology. Now, diploid, 2N, 2 copies of each chromosome. At some point, for this system to work, we had to have a stage that had 1 copy of each chromosome. That is called haploid, 1N. Okay, here's a side note. Don't get too hung up on this. Technically, haploid means half ploidy. We'll talk about plants. Plants are kind of funky. Plants can be polyploids. They could have like 4 or 6 or 8 copies of genome in there. So their haploid stage is technically half. We're going to focus on the human version of this system for the moment. So diploid, 2 copies of each chromosome in there. Haploid, 1 copy of each chromosome in there. In humans, the gametes, the sperm and egg, are haploid, 1N. During sperm and egg fusion, 1 + 1 = 2, and we end up with the 2N zygote there. Okay, one other key piece of terminology here. We've been talking about gametes here. Those are sex cells that are haploid. They're produced by a special type of division. Other cells, non-reproductive cells that are diploid, we call those somatic cells in animals. So let's look at the human life cycle, okay? Hopefully you understand this process, but we're going to go into excruciating detail here. So we have the paternal lineage here, and we have the maternal lineage here. In the ovaries, in the maternal lineage, we produce egg cells. In the paternal testes, we produce sperm cells. These are produced by a special type of cell division called meiosis. We'll talk more about meiosis later, but meiosis divides the ploidy in half. So we go from a 2N diploid into a 1N egg or a 1N sperm. When sperm and egg fuse in the process of fertilization, we recapitulate that diploid and we end up with 23 chromosomes times 2. That's a total of 46, right? So we have 23 different versions and we have 23 different chromosomes and 2 versions of each in the diploid zygote. That goes through cell divisions called mitosis and the process of development to recapitulate the mature organism here. Okay, why do I break this down? Well, you think you have a handle on how reproduction works, but I'm going to say that when you go to plants, things get different. Things get interesting. Things get really cool. So that process of meiosis, you may have learned that meiosis creates sperm and eggs. Well, in animals, that's true, but in plants, that's not actually the case. Plants have this weird thing called the alternation of generations. In animals, we have the multicellular diploid. That's you, me, any other mature human being, right? We are multicellular and we are diploid. We have 2 copies of each chromosome in our cells. In plants, they have a multicellular diploid. We call that the sporophyte. The sporophyte goes through meiosis to create these things called spores. Spores are 1N cells. Now, plants do something different here. Instead of making a sperm and egg here, they make spores. The spores divide by mitosis, and in plants, there is a multicellular haploid stage. Multiple cells that are 1N. Think about that. That's like your sperm or your egg going, dividing, and technically living its own life, being a different generation. That's kind of funky, okay? That's different, right? Those mature gametophytes, as they're called, go on to make the sperm and egg, which are made through mitosis because they're already one N, and then fertilization will occur and the gametophyte gets recapitulated. Now, in early plants like mosses and things, that mature gametophyte, that thing that is 1N multicellular, that's actually the dominant life phase. But as plants evolved, they switched over to the sporophyte being the dominant life phase. So why do I mention this? Well, because reproduction is far more diverse than you think when you think animals, right? Animal reproduction is boring, right? In flowers, we have hermaphrodites. We have male and female organs. Flowers can fertilize themselves. They can literally have sexual reproduction with themselves. That can lead to interesting evolutionary outcomes. It's both good and bad, right? If you can't find a mate, that's good backup. But we would love to have outcrossing to get new genes, new alleles in there. The system that I studied, heteromorphic self-incompatibility, combines self-incompatibility, the ability not to fertilize yourself, with different what we call mating types. So here we have both male and female organs, but they're arranged differently than in the other version. So this is the pin mating type, where the female is high up and the male is down low. And in this one, the male is high up and the female is down low. That's called the thrum. That's super cool, right? Like, that's completely different. So I come from the world of plants, where basically any reproductive system you can imagine can happen. Why do I mention this? Well, when we think about reproduction, think about humans. And we are very, very focused there on sex chromosomes, right? Here is an example of what we call a karyotype. A karyotype is when we take a picture of all the condensed chromosomes. So during cell division, the chromosomes condense down and become actually visible under a microscope. We'll see these in the lab. Humans, we have 23 pairs of these homologous chromosomes, one from the paternal and one from the maternal lineage. Each of these chromosomes has genes on it, right? And the paternal and the maternal has the same genes, but they might have slightly different versions of them that we call alleles. Okay. Now you'll notice I have two karyotypes here. One of them has something different in it. You're probably looking at the X and the Y chromosome. That is a difference, okay? We do have these things called sex chromosomes, which we use to determine male versus female, although that line is not exactly clear all the time. But you might also notice there's 3 of 21 here. You may have heard of trisomy 21, Down syndrome. So we use karyotypes to look at a variety of things, and we can often see sometimes large-scale changes in the number of chromosomes can lead to outcomes like, in this case, trisomy 21, Down syndrome. So let's talk terminology here, just to be clear. We have homologous chromosomes. In the diploid cells, these are the matching pairs of chromosomes. So these are homologous chromosomes. Chromosome 1 from the paternal lineage, chromosome 1 from the maternal. Okay. A gene is a region of DNA on a chromosome that codes for an RNA. We've seen that definition before. That is our very specific definition for this course. Don't use any other definitions. An allele is a different version of the same gene, right? So an example would be ABO blood type. You can have different versions of the same gene that lead to different blood groups, as we call them. We might have alleles for flower color. We're going to talk a lot about flower color when we talk about Mendel and genetics. Okay, so back to the karyotype. This is a biology course. I'm going to teach you the biology, but we don't understand all the biology. And biology intersects with society and concepts of our society. We're humans. We can come up with all kinds of constructs. We've come up with money and cars and all these different things, right? The biology here, right, when we look at a karyotype, we see X chromosomes and Y chromosomes. We equate that to sex, right? Male versus female. But the karyotype doesn't tell us everything about a person. It doesn't tell us about how the genes that are on there are actually expressed. So we see different ranges here, right? And there is a continuum that is in here, and we are constantly learning more and more about this. We do see 2 local maxima that we tend to call male, well, in this case, female and male, right? But so here we have the biology of it, There can be differences in the expression of these things, right? It doesn't tell us everything. We also have the societal concepts of gender. This is something that we're not going to talk too much about in this course because I'm here to teach molecular biology, right? Not sociology or psychology or any of these things that are there. But they are important components to our society, right? And they're constantly being discussed right now. So we are going to focus on the biology here. I will use terms like male and female to describe this, but that has nothing to do with gender. Also, I will use things like maternal and paternal lineage to describe where the genetic material is coming from. That doesn't fully reflect people's social and family lives in how complex they are, right? So, human life cycle, long story short, is kind of boring, right? When we compare it to something like plants, where plants can have sex with themselves, with other things. They can have weird numbers of chromosomes, right? Don't even get me started on fungi. Fungi do weird stuff, right? So we are going to talk a little bit about human reproductive biology and these divisions here, but the systems work differently in different lineages of life. Bacteria don't even do sexual reproduction, so their genetic diversity is completely different. All right, let's finish up by talking about DNA organization. So you saw those chromosomes tightly packed in the karyotype. Well, how do they get like that? Well, when cells divide, the DNA becomes tightly wound, and that can actually affect whether the genes are on or off at that point. So your genome in each cell is about 2 meters long. That's over 6 feet. That's a lot of DNA in there. How do you pack all of that into the nucleus of each cell? Well, this is where a series of proteins come in. We have things like histone proteins. DNA gets wound around these histone proteins. That helps wrap it up. That forms a nucleosome, and then these further coil and condense, forming what we call chromatin. Chromatin is a mix of DNA and protein. It's the complex of those two things. That gets coiled and condensed and wound tighter and tighter. We can have different states of this. We would call super tightly wound DNA heterochromatin. Heterochromatin is often not very active. On the other hand, more loosely wound DNA we would call euchromatin. That can be active. The genes can be expressed there. So when we see our duplicated chromosomes, like in the karyotype, that would be heterochromatin. And when we see cell division, we see the X-shaped things. Those are sister chromatids joined together at this place called the centromere during replication. That only happens very, very rarely. Okay, so we have the genome, right? All of the DNA within a cell. Prokaryotes have a single circular chromosome, might have some extra circular plasmids in there. In eukaryotes, we have multiple linear chromosomes making up our genome. We have to talk about ploidy. How many copies of each chromosome are present in a eukaryote cell? Most of our cells are diploid, right? We have 2 copies of each chromosome, one from the maternal lineage, one from the paternal lineage. A haploid cell has only 1 copy of each chromosome type in there. In animals, we see sperm and egg cells being haploid. Those are our gametes. In animals, the gametes fuse together. 1 + 1 = 2. We get the diploid zygote again. During cell divisions, chromosomes condense, and you can even see them on a microscope picture. We call that a karyotype. The homologous chromosomes are pairs of chromosomes. We called it chromosome 1. We had 1 maternal and 1 paternal. Those are the homologous chromosomes. Genes are regions of DNA on a chromosome that code for an RNA. An allele is a different version of the same gene. You might inherit one version from the maternal lineage and one from the paternal lineage, or they could be the same. DNA is wound around histone proteins to form chromatin. Euchromatin describes less tightly wound DNA, and heterochromatin is very, very tightly wound DNA. Okay, in the next section, we'll talk about the cell cycle.